Welcome. Thanks for joining us. Thanks for joining us this morning for our very special McCance seminar from our clinical trials director, Gene Bowman, also director of the Brain Nutrition Lab. Uh, Gene joined us a couple of years ago as a clinical trials director. He's also, we first met when I was on the SAB at Nestle Health Institute, and Gene was running many of the medicinal food and clinical trials there. Gene's been in the field for over 20 years in nutritional neurology, focusing on nutrition, cognitive aging, and Alzheimer's disease. He received the NIH Ruth L. Kirstein Award for his work on cognitive impairment related to gastric acidity. And he has many publications, including one I'm sure we'll hear about, a trial using omega-3s and PUFAs for treating Alzheimer's and dementia. He also co-founded the AAI Start Nutrition, Metabolism, and Dementia Group, which maybe he'll mention, and is involved with multiple clinical trials. His work has been featured widely in the media. He really is truly a leader in the nutritional aspects of brain health, and we're really happy to have him in our center playing these two roles, both in nutrition and brain health and running our clinical trials. So with that, I will let Gene take it over, and we look forward to your talk. Gene will be talking about targeted nutritional therapies in aging and Alzheimer's dementia. Take it away, Gene.
Okay, thank you very much, Rudy. Thank you all for attending the seminar. So I'm going to tell the story here a little bit about the role of diet and nutritional metabolism in Alzheimer's disease and cognitive health. So we're going to talk a little bit about food and dietary patterns, but a lot of it is going to be based off of molecular biomarkers that we've developed that objectively assess nutritional status and how we take this approach to better target nutritional therapies for prevention of cognitive decline. These are my disclosures. So this is just a bird's eye view of what's going on at the McCance Center from a clinical research standpoint. As Dr. Tanzi mentioned, on the right side, you see these laboratory models of brain disease and the 3D organoid models, brain organoid disease models that Rudy and the Genetics and Alzheimer's Research Unit have developed that have put forth leads that need to be translated into early proof of concept clinical trials. And we'll talk a little bit about that from a high level. One of the designs of those proof of concept trials is at the bottom here, where we have serial measures within person over time, pre and post treatment, to detect early effects of these potential new combinations for preventative and early treatment in Alzheimer's disease. And the majority of what I'm going to focus on today is on the left side, is really the approach that I've been developing over the last couple decades, utilizing nutrient biomarkers and analytical chemistry to measure nutritional status both objectively and reliably, to understand the delivery of nutrients into the central nervous system through a host of different mechanisms at the blood-brain barrier and the choroid plexus, understanding that there are subtle changes in the blood-brain barrier function as we age, particularly as people move into an Alzheimer's dementia. Of course, in vascular dementia, it has a more paramount role, but disturbances in the transport of nutrients and nutrient delivery to the brain. And then how we approach the analysis of molecular biomarkers in the setting of cohort studies of brain aging is not so straightforward. So it's utilizing a brain discovery biomarker platform, but using dimension reduction techniques and understanding nutritional biochemistry and combining certain nutrients in certain ways that reflect pathways. And through this work, it's led to a couple clinical trials. Our clinical trials platform is really focused in three areas around consumer health. What are the things that are already available that we could package in such a way that could help people take a proactive role in prevention of cognitive decline? And then more regulated paths of medical nutrition, developing medical foods, and then also drug development and combinations thereof. So this slide is one of the most popular slides, I think, over the last 20 years. But it really is important to understand how you set up a clinical and translational research program leading to formal clinical trials. So we understand the natural history of disease development in dementing illnesses, particularly Alzheimer's disease, that really begins with amyloid beta accumulation, reflected here from the preclinical to MCI to dementia stage. In each of these stages, there are profile changes in molecular biomarkers, whether it's neuroimaging, cerebral spinal fluid biomarkers, and now in the blood, that appear to become abnormal or disturbed as the progression of the seeding of the disease begins decades before the onset of clinical symptoms. Now, from a clinical trial standpoint, there's really three areas that we think about and conceptualize this. It's a primary prevention paradigm, which has been very difficult in the field up until more recently, where you're trying to delay the onset of Alzheimer's disease pathology from the very beginning. Then there's the secondary prevention, and this is where the majority of the field has moved to delay the onset of cognitive decline in adults with risk factors. So they have pathology, but they don't have the full-blown dementing diagnosis of Alzheimer's disease, or they have small vessel disease or energy deficit, some type of risk factor where there is biomarker evidence of disease developing. And then the majority of the trials have been in the tertiary prevention or treatment stage where you enroll people with mild to moderate Alzheimer's disease and trying to delay the progression of the dementing illness. And you can see that at that point, you have all the aspects of the abnormal features that have developed, both not just the clinical disease and diagnosis, but also from a biology standpoint, the majority of the pathology is already set in, and it may be too late to identify any helpful treatments at that point. Now, thinking about some of the pathogenic mechanisms that are involved in cognitive decline in Alzheimer's disease, but from the point of view of nutrition, metabolism, and cellular nutrition, I think there are particular key areas that we've published on that nutrition plays a dramatic role in the direction that the biology goes. Whether it's cerebral energy deficit, the classic hallmarks of the disease, aggregation of tau and amyloid, the epigenetic expression in the nourishing nutrient pool within the cells can be modulated by changing certain nutrients. Synaptogenesis, at least in a dish, and partially in human evidence, can be modified through dietary changes or nutritional changes in a dish or the media that's in the dish. Neurovascular dysfunction, so this is breakdown of the blood-brain barrier or delivery of the nutrients and oxygen and water and so forth through the central nervous system. And then inflammatory pathways. Nutrition has a paramount role, not just in the central nervous system, but certainly in many organ systems from promoting or downregulating inflammatory expressions. So in cerebral energy deficit, we, through the Alzheimer's Association iStar, the Nutrition Metabolism Dementia PIA, an international cadre of people, including Dr. Tansey, but people all over the world came together to understand better how cerebral bioenergetics are influenced through nutrition in the setting of Alzheimer's disease and related dementia. So I point you to this direction. We've focused on key nutrients, lipids, and micronutrients that have a cofactor role in promoting bioenergenesis in the brain, mitochondrial function, and also lipid metabolism that potentially could be utilized to promote, for example, medium chain triglycerides. We've also done some work related to the classic hallmarks of Alzheimer's disease using cohorts that we had readily available. So this is a study where we developed an assay in the CSF and in plasma that maps out the one carbon pathway and looked at how that related to the levels and concentrations of the one carbon pathway biomarkers in relation to classic hallmarks of Alzheimer's disease. So CSF levels of p-tau 181 and total tau, And you can see here that acetylhomocysteine in the CSF, which is here, a metabolite of homocysteine, is related to elevated levels of p-tau 181 in living subjects, whereas 5-methyl tetrahydrofolate, the active form of folic acid, folate, in the CSF is inversely related to p-tau 181. And it's even more dramatic if you look at total tau in the central nervous system of people at risk for dementia. And we see an interesting interaction with ApoE4 carriers. For example, plasma homocysteine, which is here in the pathway and is lowered with vitamin B6, folate, and vitamin B12. So elevated plasma homocysteine levels is associated with elevated CSF p-tau 181, but only if you're an ApoE4 carrier. Plasma homocysteine levels don't seem to be relevant to the central nervous system in non-carriers of the E4 allele. So this is some of the work we've looked at, some of the pathways that are linked to diet and nutrition related to Alzheimer's disease pathology. And then, of course, getting into the neurovascular dysfunction and inflammatory pathways. Earlier, when I was a postdoc at Oregon Health and Science University, my first couple of years learning about the disease, both clinically and also in research, it was quite controversial to say that there's blood-brain barrier dysfunction in Alzheimer's disease. In fact, we try to remove that input in terms of risk or progression in our clinical trials using the Hitchinski ischemic scale, where we exclude people that have some kind of sign of some vascular component. But these are a couple studies in review on the left that we've published in a very well characterized mild to moderate Alzheimer's disease group that came to autopsy at our Alzheimer's Center in Oregon, where we see that the CSF albumin index, which is a measure of blood-brain barrier integrity, is strongly related to annual CDR sum of box change. So it's clinically relevant, but it's also related to elevated risks of cognitive decline, where you see the CSF albumin index here on the left side, on the y-axis, related to CDR. So it appears that those that are cognitively intact with a CDR clinical dementia rating of 0 have a lower CSF albumin index, which indicates less blood-brain barrier breakdown. But then when you get into the MCI and Alzheimer's disease, and of course, we don't have a lot of cases in the AD, it appears that the changes have already taken place. There's already a nice bump in breakdown of the blood-brain barrier, even at the MCI stage. And we've identified some inflammatory biomarkers that really explain the CSF albumin index in mild to moderate Alzheimer's disease, and they all are related to vascular injury. So even in people that don't have dementia, you can see that there is some subtle change, even with a crude measure of CSF albumin index, looking at the ratio of serum to CSF albumin. Albumin really shouldn't be getting into the central nervous system. It's a large protein, but as it begins to leak into the central nervous system, we can capture that, and that will give us an index measure of blood-brain barrier integrity. Now, this is not addressing any changes. Now, this is the breakdown in the tight junctions, things leaking into the central nervous system or spilling. Permeability is increasing. It's not a measure of transport systems. Just want to make that clear. So this has been replicated using different like K-trans, where you infuse compounds and you can measure the transport into the central nervous system, and others have shown that as well. That's very consistent with what we see with the CSF albumin index. Our study here in Montane's neuron paper in 2015, and some of the mechanisms around inflammatory measures that we identified earlier in the earlier study. So I'm going to transition. So that's just a little bit of a highlight of how we start thinking about the role of nutrition and natural bioactives and nutrient delivery to the brain in terms of the pathology leading to Alzheimer's disease. But if we back up a little bit, in the Alzheimer's field, it's a little bit more challenging to study nutrition because the measurements themselves have been subjective in nature, where we're asking people the frequency of foods that they're eating, And you want to do that. You want to use a food frequency questionnaire to understand what people are eating. It does give a representation of the frequency of particular foods that are eaten that correlates with the volume, that correlates with a specific amount that they're receiving each day. And it taps into generic memory, which is a little bit easier task than episodic memory. So we're not asking, okay, what did you have to eat last night? We're asking the frequency of consumption of foods over time. So using the food frequency questionnaire, if you're going to use a subjective measure, is the best way to go. But of course, it succumbs to measurement error through recall bias. We've done some work to look at the relationship of dietary questionnaires in relation to what's circulating the bloodstream and thresholds that are in the bloodstream that really are related to neuroprotection or increased risk for cognitive decline. So this is one pilot study that we conducted years ago now, but I'm just going to introduce it for maybe younger scientists that are here trying to think about how to get into the field and how to really build a hypothesis around nutrition and metabolism linked to brain aging and cognitive decline and Alzheimer's disease. So in this study, we enrolled 38 people, half with MCI, mild cognitive impairment, and half age matched that were normal, cognitively intact adults. And we had them come in at baseline, fill out a food frequency questionnaire, have a blood draw for nutrient biomarker panel and cognitive examinations, and had them come back 1 month later and redo everything. And the reason for that is because, a month of time is enough time to is short enough where people don't change their diet. So what they're reporting on the food frequency questionnaire should be exactly the same. And then it's also short enough to where you can complete the study and look at test, retest reliability, both in people with MCI versus people that are cognitively intact, or how reproducible it is regardless of their cognitive status over time. And so these are the biomarkers that we measured that were also reflected on the food frequency questionnaire, included vitamins and minerals and lipids. Okay. And if you look at the vitamins, ascorbic acid, vitamin C, vitamin E, different forms of vitamin E, lutein and zeaxanthin, some of the carotenoids and xanthophylls and the B vitamins, this is a correlation, just looking at the correlation between what's reported on the food frequency questionnaire and what you see in the nutrient biomarkers. And you should see a positive correlation coefficient here. And for some of the nutrients, it's quite strong. So the breakdown of what people are eating to the nutrient database to then push out numbers for that individual of what vitamins and so forth they're eating, the carotenoids are quite reproducible and reliable. So those do a pretty good job with relatively high coefficients. But the others, you see some negatives are quite low. For the minerals, for example, you see them in the opposite direction. And so for some of the lipids, you see them in opposite direction. However, when you stratify the population and run the analysis in MCI, so people that have cognitive impairment versus those that do not, you see that a lot of the negative coefficients and you lose a lot of the significance for any correlations. It gets much worse if you limit it to just the MCI group. And it gets much better if you just limit it to the cognitively intact group. Okay, so it's clear that something's happening in the MCI group in terms of what they're reporting. They're probably reporting what they ate when their memory was intact, not actually what they're eating currently. So if you look at the reliability statistics over time in people with MCI, you see that what they report on the food frequency questionnaire is quite reproducible at baseline and then 1 month later, but it's not accurate. Okay, it's not what is circulating in their bloodstream. Okay, whereas people that are cognitively intact, it is the case. So these are just some additional correlations that you would expect to see between plasma vitamin B12 and lower homocysteine, plasma folate, and plasma homocysteine. So higher levels of folate, higher levels of B12, lower homocysteine levels. But with the dietary reflection of those measures, there's no relationship with homocysteine. This is looking at the reproducibility. These are intraclass correlation coefficients. The longer the bar, the more stable it is over time. And so this really dispels the myth, in nutrition science at least, and we're in a neuroscience crowd here, but in nutrition science, the dogma, the standard has been that a lot of the nutrient biomarkers are unreliable. They don't reflect very well, and they don't correlate very well with disease outcomes. I think we dispel that myth here with these data, and of course, over the last decade or more now, that if you do a good job with collecting and handling the biospecimen and with state-of-the-art methods that are available in analytical chemistry and mass spec now, we can do a terrific job of getting reproducible interclass correlation coefficients here. You can see most of them, the dotted line here is plasma cholesterol, which is a clinical benchmark. A lot of them are better than cholesterol. Some are worse. You do see heterogeneity. Some do not perform well, and there's reasons for that. Vitamin C, for example, is lower in terms of reproducibility because you have to have an additive added to the sample to preserve the vitamin C in the specimen. And if you don't do a good job of that, then you get a lot of variation in the measure, and that can affect the hypotheses that you can pursue. And if you just look in a clinic population, so this is data from a clinic population that we're seeing at the Oregon clinic, at least, of 40 people. And across the top here, if you just use an Excel spreadsheet and you look at each individual's nutrient profile, you can see the B vitamins, the orange is denoting an abnormal level. And you can see for the B vitamins, we do a pretty good job in clinic because it's part of the standard of care. You look for reversible causes of dementia, which include vitamin B12. And you see very few people actually that have insufficient levels. But if you look at some of these other nutrients that have been implicated or lipids, you see that a lot of people have suboptimal levels. So I think there's opportunity here to identify, the nutritional requirements and metabolic profiles that are really significant. And then if they become part of standard of care, then we do a really good job of intervening and capturing that. But right now, this is where we stand, and we have a lot of people with suboptimal nutritional status coming into clinic. So moving forward, now that we had these nutrient biomarker methods updated and validated, we took them to a larger cohort called the Oregon Brain Aging Study. We measured 30 nutrient biomarkers here, including the B vitamins, antioxidants, the carotenoids, vitamin D, trans fatty acids, omega-3 fatty acids, omega-6 fatty acids, and lipids. And this was in a cohort of 104 people with a mean age of 87. So these are all non-demented individuals. We applied principal component analysis to the nutrient biomarkers. And these are what we call the nutrient biomarker patterns. From a statistical standpoint, these are each principal component that is extracted using this model. So PC1, 2, 3, 4, 5, all the way to 8. Now we label these as nutrient biomarker patterns because that's essentially what they reflect. So pattern number 1, highlighted here, means that each of these nutrients have high correlation coefficients with this pattern. Okay, so we can scale each individual on pattern number 1, which means that they have higher levels of vitamin B6, B1, B2, folic acid, C, E, B12, and D collectively. Okay, so then we can look at how people score on the nutrient biomarker pattern number 1 relates to disease outcome here. So these are how much variance is the eigenvalues, how much variance is explained as each extraction, where you stop and how many patterns you pull out. And this is the distribution of the score. This is pattern number 1, the distribution. You see a nice distribution of that score. And to make a long story short, so these are some of the findings that we had. So for example, the BCDE pattern number 1 in the bloodstream. So those that had higher levels of B, C, E, and D in this cohort had larger brain volumes. Whereas those with higher pattern number 1 did not have any significant relationship to white matter hyperintensities or as a surrogate marker of cerebral small vessel disease. However, the omega-3 pattern, looking at EPA and DHA that we identified, was not related to total brain volume at all. in our hands, but it was strongly inversely related to white matter hyperintensity volume. So almost 25% of the variation in white matter lesions in older adult brain is explained by EPA and DHA concentrations, which was a new finding at the time. And this is a summary of all those findings where each pattern is related, associated with a particular cognitive domain and also brain MRI. So the BCDE pattern was not related to memory, language, or processing, but it was related to these other domains. Global function, if you collapse them all into a cognitive Z-score, and it was also associated with higher total brain volume. Saturated fats, carotenoids, and cholesterol did not have a significant effect that we were able to detect in this study. The omega-3s were associated with better executive function and also less white matter lesion. The omega-6 appeared unfavorable. Trans fat was the most unfavorable, and lutein plus zeaxanthin plus high HDL was the only one that was related specifically to memory recall. And all of these models were adjusted. Now, of course, these were observational results, but we at the time were intrigued by this result, given that we have known for some time as people begin to lose their executive functions, that the underlying biology there is probably related to small vessel disease in some form or fashion. So to see that the omega-3s were both related to white matter lesions and also executive function made us think that this might be causal in some form or fashion. A lot of people have questioned, the magnitude of the effect on nutrition, and I just wanted to put this up there that if you looked at global cognitive function in this cohort, so if you just look at global cognitive function as a Z-score of all of these cognitive domains combined, age, gender, education, ApoE genotype, hypertension, depression explains 46% of the variance in global cognitive function. If you add the nutrient biomarkers to that, you can add an additional 17%, leaving 37% unknown. If you look at total brain volume, it's even more compelling, where the same covariates explain about 40% of total brain volume. If you add these nutrient biomarkers, you can almost double that. Okay, so about 77% of the total variation in total brain volume is explained by these known risk factors and nutrient biomarker patterns. So that begins to kind of give, I think the methods that we're using, because it's closer in proximity to the central nervous system, and we're able to accurately and reliably measure these nutrients and concentration, the dynamic flux there, that we're able to identify new results that with a diet survey were never possible. So we were fortunate to get NIA funding to conduct an omega-3 trial in a similar population, age 75 and older, that were non-demented over 3 years. And we required that people had white matter lesions that we had shown that seemed to be linked to the omega-3 concentrations. So they had to have significant white matter lesions, but be non-demented, and they had to have suboptimal omega-3 status. And we followed them over 36 months. The main outcomes were 3-year change differences between the groups, and MRI total white matter hyperintensities is the primary outcome, and diffusion tensor white matter integrity, medial temporal lobe as secondary outcomes. We pre-specified ApoE genotype stratification, and we had biomarker and pill counts for adherence. We published the results of that trial in JAMA this summer, and it was a negative trial in that the omega-3s at 1.65 grams over 3 years did not seem to modify white matter lesion change over time. But we did see a signal with slowing the decline in neuronal integrity with the fish oil that was concentrated in omega-3s, EPA, and DHA versus the placebo. Then when we so this was a p-value of 0.06 in all participants. And then when we stratify by ApoE genotype into ApoE4 negative participants, we see really no results there. But in the ApoE4 carriers, we saw that the fish oil as early as 1 year began to, those that were randomized to the fish oil versus the placebo, appreciated slowed breakdown in white matter integrity as early as the first year, and that was maintained throughout the entire duration of the 3 years. So I think that was the key finding from this phase 2 trial. And this is just highlighting that a little bit deeper, where we see clear effects in ApoE4 carriers. Um, Now, some of the other nutrient biomarkers, so we've had discovery projects where we have some would call it agnostic or data-driven approaches where we select biomarkers that are reliable and robust that are also, we think, related to certain pathways that should be disturbed in people that are developing an Alzheimer's dementia. Once we identify those, then you have to think about, we can't measure everything. So what are the key ones to focus on? Which ones have the biggest impact? And which ones are particularly synergistic, but also reflecting separate pathways that if we have a measure, a marker of a particular disturbed pathway and a second pathway and a third pathway, will that be able to um, be clinically relevant? And so around that concept and thinking, we conducted another study in the multi-domain Alzheimer's prevention trial, where we selected just EPA and DHA, the omega-3 fatty acids, vitamin D, 25-hydroxyvitamin D, and plasma homocysteine. And in that trial, we selected 25% of the population across each arm. This is the parent trial on the right. It was a null trial of 1,680 people, followed over 3 years, that had a multi-domain intervention with and without omega-3s. And they reported null results across the board. So we went into the baseline samples, 25% of the population in each arm, and we measured these nutrient biomarkers and created an index score. So for each of these, we gave each individual a 0 or 1 based on the concentration. Now, how did we select the concentration? So for EPA and DHA, the omega-3s, they did it for us in this trial. They looked at the effects in those below or above this omega-3 level. So we kept the same, and we pre-specified that. The 25-hydroxyvitamin D is set at the clinical deficiency, the well-established clinical deficiency criteria. And homocysteine level was based on Shashandri's paper in the New England Journal of Medicine years ago in the late 90s at the Framingham. So we utilized those as the cutoff. The score of the construction of the nutritional risk index, which we call this in terms of asking the individual the question, are they or are they not at nutritional risk for cognitive decline? Okay. So the score goes from 0 to 3 with higher scores representing increased nutritional risk. Okay. So those for the omega-3s that they had less than 4.8 weight percent of total, they received a 1. For the vitamin D, if it was less than or equal to 20, they received a 1. And for homocysteine above or equal to 14, they received a 1. The total population was 712, mean age is 75. They were all non-demented at baseline. ApoE4 carriers, about 20% of the population. And these are the concentrations of the nutrient biomarkers, which look reasonable. Homocysteine is much higher in France. This is a French study, much higher in France than it is in the US. They do not have fortification of grains with folate or homocysteine modulating compounds like we do in the US. So that's one thing to keep in mind. When we looked at the rates of change in cognitive function using the same primary endpoint that they used in the clinical trial, but now looking at the differences in the nutritional risk index at baseline, these are the results we saw. So those that had no nutritional risk or optimal nutrition, an NRI of 0, are the ones that actually had the learning effect on the clinical trial over time. So they improved on repeat testing their cognitive scores. As soon as somebody had one of them were suboptimal, they lost that learning effect. So it could be any one. It could be due to homocysteine, vitamin D, or the omega-3s. Okay. Once you had 2, then you had clear cognitive decline compared to optimal. And then if you had 3, even at baseline, you were well behind anybody in the NRI of 0. So this is suggesting that there's incremental effects as nutritional risk increases, so does the rate of cognitive decline. Now, if you just look at, the omega-3s, the vitamin D or the homocysteine, and which one's contributing the most, it's not really clear in this study, but maybe the vitamin D here was the most relevant, whereas those that had an NRI of 1 of vitamin D had significant decline, where the other two, if they only had either omega-3 or homocysteine, didn't seem to be significant in reference to optimal nutritional status of NRI of 0. But when you get into 2, it didn't seem to matter. All combinations were relatively significantly worse compared to the reference group of no nutritional risk. So French elders with a homocysteine less than 14 micromoles per liter, vitamin D greater than 20, and red blood cell PUFA greater than 4.8 appear to sustain cognitive function with aging over 3 years. So are these thresholds really important? Are they neuroprotective thresholds? Still an open question, but that's what these results support. These findings require confirmation in U.S. population post-folate fortification era. So these are the conclusions that we drew from that paper that was published in 2019. The 3 nutrient biomarkers can be expanded to offer more comprehensive biomarkers of diet quality in relation to brain health. We only chose 3. Gene-nutrient interactions should be explored to assess relevance to optimum concentration thresholds. So, for example, if you're carrying the ApoE4 allele, should your threshold be different in terms of neuroprotection? And identifying nutrient combinations in observational studies might be further explored in vitro using, for example, Dr. Tanzi's models. So lo and behold, this summer, a Framingham heart study came out where a group from Wageningen University and Sudha Seshadri's group in Texas looked at the nutritional risk index in relation to all-cause dementia incidence, and they found that homocysteine, vitamin D, and omega-3 using very similar methods and index score saw a 4.3 to 4.7 fold increase in dementia incidence in those that had the highest nutritional risk index. So this is a separate group, U.S. population, that has identified that these 3, when they are suboptimal, is also associated with significant risk for dementia in the Framingham Heart Study. This is also some cell culture models that confirm the synergistic properties of the omega-3, EPA, DHA, and vitamin D. So this is work that was done in the Genetics and Aging Research Unit, showing that each of these has independently, each of these compounds can reduce TNF-alpha concentration or expression or release from microglial cells. And when you begin combining these, you can see increased magnitude of effect on those dampening effects. So we have also in vitro support of these combinations being relevant and having additive effects. And so this led to a proposal just recently that we submitted in October for targeted nutritional therapy for dementia prevention in middle to late life age in people that have the highest genetic risk and nutritional risk that we know of. So it's people targeting age 50 plus that are ApoE4 carriers and they have a nutritional risk index of 2 or higher. And we will intervene and follow them for 24 months. And the main outcomes are going to be blood-based nutritional risk index. So we want to make sure the formula, the supplement we're giving is optimizing the nutritional risk index. Then we want to see if we're having effects on the biology using neuroimaging, diffusion tensor imaging, fractional entropy, that we saw the effects in the ApoE4 carriers with the omega-3 is one of the primary endpoints. Also medial temporal lobe cortical thinning, and some blood-based AD biomarkers. This is a secondary analysis in the Framingham Heart Study that we're putting together to understand how the nutritional risk index is related to Alzheimer's disease biomarkers that are available, including A beta 42 to 40 ratio, p-tau 181, NFL, and GFAP. So we have some preliminary studies, but we want to carry this through and look at change over time as an addendum to that grant, and also to understand the mechanism, how this nutritional risk index is associated with such a higher risk for dementia in the Framingham. Is it related to Alzheimer's disease mechanisms in that cascade, or is it other mechanisms? So, of course, outside of homocysteine and omega-3s and vitamin D, there's still a lot of other nutrients that may have a role. We have the nutrition and brain aging study in the brain nutrition lab at the McCance Center that has 154 nutrient biomarkers from amino acids and minerals to fatty acids and liposoluble and hydrosoluble vitamins. And we'll be looking at how those relate to global and domain-specific cognitive measures and neuroimaging parameters, and also postmortem brain tissue analysis, CRAD, and Brock staging. And we have a paper in development looking at the long-term intra and inter-individual reproducibility of those. So we have about half of the population of 315 that have repeat measures, where we're looking at reliability of the nutrient biomarkers over time. And then we also have a review paper that's in development where we have reviewed all of the dietary patterns that are associated with cognitive health and Alzheimer's prevention, including the MIND diet, the DASH diet, the Mediterranean style diet, the modified Mediterranean ketogenic diet, the Nordic diet, Eat Lancet, USDA dietary guidelines, looking at different food groups from green leafy vegetables and berries to fish and whole grains to poultry, potatoes, wine, and alcohol to sweets and fried foods. And we've compared each of these, and actually there's quite a bit of heterogeneity in terms of the recommendations for each of these diets. And it turns out that there may be a new dietary pattern that has yet to be tested formally in a clinical trial that may take the best features of all of these diets. Not that somebody would be able to adhere. So, for example, to 5 servings per week of fish, for example, or 15 to 18 ounces per week of fish may not be reasonable, but with some supplementation. What we're probably looking at is maybe a novel neuroprotective diet that we would hope people would adhere to at about 60 to 70%. So anyhow, that's what else is going on in the lab. So future directions. So we've identified key nutrients and combinations associated with dementia risk. More comprehensive nutrient profile is in development. We can now identify people at risk for future cognitive decline using a blood test that assesses nutritional status. Omega-3 beneficial effects are amplified in ApoE4 carriers. There may be a novel dietary pattern for brain health that has yet to be formally tested. The McCance Center is establishing blood nutrient concentrations required for optimal cognitive health. It's developing the nutritional risk index as a tool to predict risk for dementia based on nutritional status, and is integrating the NRI into nutrition-based clinical trials for the prevention of Alzheimer's disease. So with that, thank you all for your time, and I'd be happy to field any questions.
Great. Thanks, Gene. Very informative and fascinating talk. And you can open this up for questions. You can just speak up or raise your hand, whatever you prefer. So questions? I can ask the first one. Gene, so if I was to have the nutritional risk index test and I was lacking in 2 out of the 3 main components that you talked about, is the recommendation change your diet to get those components, or would you put somebody directly on supplements to up those components? Like, what would be the protocol?
Yeah, I think we need a variety of options for people. I think it depends on the individual. So if this is somebody coming into the clinic, that is able to change their diet and is very open to that, and that's very clear. I think there's an opportunity for running that test and giving dietary guidance on how to lower their nutritional risk for cognitive decline, and maybe following up in a month and rerunning the test and see if they've been successful. If they have not been successful, then I think you're moving into a supplementation regimen with the idea that these are hazardous levels of nutrients that the evidence continues to build that are placing people at remarkable risk. And what's the harm in changing their diet or removing certain things that may be increasing that hazard? So I think you need a little bit of both. I think it's possible to do it through diet, and we have dietary patterns and regimen that would do that. But whether somebody's able to do that and implement that in their life requires some flexibility and the idea of maybe some pill pushing in terms of supplements that would also do the job.
I guess if somebody was dramatically low, you might want to use supplements first and then start working toward modifying their diet once they're stabilized toward those.
That's right. That's right. That's a good point. I mean, if you have somebody that is, clinically deficient in these nutrients, you may even require some injection, um, routes.
Yeah.
So for example, vitamin B12, we've done that in the past for people that have very
Mhm.
low levels. Do a gluteal injection for a week or two to get people back on track.
So Drew has a question before so one last question, and then I'll hand over to you. Any correlation between NRI and I've been a vegetarian since college, but is there a problem with NRI, particularly in vegetarians, because of lack of B12 or meat? I mean, I supplement, but some people don't.
Yeah, yeah, yeah. So that would be caught on the homocysteine portion of the test for the NRI.
But do you see any correlation of a bad NRI with vegetarians, or you never looked at that?
Yes, there are. Well, we have not done that, but there's an established relationship with vitamin B12 that
Yeah.
serum levels of vitamin B12 is lower in strict vegetarians.
Mhm.
The primary sources are animal products for vitamin B12. But outside of that, for example, folate, which will also drive down homocysteine levels, is really plant-based, right? Dark leafy greens. Um, so it's really for the vitamin B12 portion.
Okay. I'll stay vegetarian for
Okay.
now. Do, why don't you ask your question?
Yeah, I'm wondering why the nutrient deficit is observed in Alzheimer's patients. And then one idea is that their diet habits are different, or the other idea is that their cellular metabolism for nutrients are messed up by the disease. And I'm wondering, is there any studies with the controlled diet and how these different alter cellular metabolism of these nutrients? And like say that if you're just looking at the blood nutrients levels, there are all differences, but they are really contaminated or confounded by different dietary habits. But if we have a mechanistic idea how certain level of nutrient metabolism is altered, and then we can actually go to these markers as a Alzheimer nutrient blood biomarker. So I wonder whether how much study has been done. Which one is really regulated by diet? Which one is regulated by altered metabolism?
Yeah, that's hard to tease apart in these studies. I mean, what we can do is we can look at certain features of pathology and understand which of these markers, these biomarkers, are explaining different aspects of the pathology
Mhm.
and hypothesize around that. Of course, it's not a completed story. So, for example, one carbon metabolism and homocysteine levels and S-adenosine homocysteine and the 5-methyl tetrahydrofolate, that one carbon pathway seems to be disturbances there,
Mhm.
seem not to be related to amyloid pathology at all, strictly to tau pathology, meaning you can't find a relationship in living participants between one carbon pathway biomarkers and CSF A beta 42 or 40, for example, but they're robust for total tau, CSF total tau, and
Mhm.
for p-tau 181. So the homocysteine and one carbon pathway may be linked to neurofibrillary tangle
Mhm.
production in some form that maybe is separate from the cascade from amyloid to tau for whatever reason. Now the in vitro work done in your lab, and Rudy, is pointing to the effects of the omega-3s and vitamin D really on microglial activation, right? Decreasing the release of TNF alpha, not on specific A beta 42 uptake. So it appears to be working more through an inflammatory mechanism. So when we look at these biomarkers, each of these biomarkers, I kind of, glazed over them, but it takes careful thought on each one of them to understand how they're metabolized and what the measure is actually reflecting. So it's an important point that you bring up. But what we do is we identify these patterns that seem to be relevant, and then within that pattern, we think about the biochemistry and how it's related to the neurochemistry and the different pathological features.
Mhm.
And we select those and do more of a deep dive, and that's what we've done for these three. But you can see there's a ton of work to be done, and it's very multidisciplinary.
Yeah, I think there's that one thing I can propose is that we can go opposite direction. It's opposite perspective. We have Matthias's brain organoid models. We can induce amyloid accumulation week 1, week 2, week 3. And as you know, these organoid has really controlled the nutrients. Everything's controlled. So if you're
Right.
looking at what
Right.
nutrients changing, happening unbiasedly, not like one,
Ah,
two, and then maybe you can connect them with some of the Alzheimer's patient observation. And at that moment, like we can go a little bit deeper into part of the things. Because right now, I think it's just nutrient deficiency index and others. It's not very helpful to say that that can be used for identifying Alzheimer's patients. But if we have something directly core nutrient deficit by amyloid accumulation or tau accumulation in model system together, and then we may actually see something predictable, right? That's all that matters. If you see
yeah.
certain type of nutrient deficit ratios, and then you can determine this is Alzheimer's patients, and that I think that's actually our potentially very exciting direction. So yeah, I'll be happy. I think that's actually a very interesting point. So thank you very much for your talk.
Yeah. Thank you for the thoughts.
Also, I'll point out, Louisa Kinti, who did those experiments on the EPA and DHA, she showed that it may be a way to reprogram microglia, less neuroinflammatory status and more phagocytic effects on uptake and clearance of A beta. So, and if you did clear A beta and have less inflammation, both of those are going to contribute to less tangles in your in AD, which could be part of what you were seeing in those ApoE4 individuals in the trial. Okay, so Martin, you want to ask a question?
So,
Hey, Martin, you're breaking up. Are you're not, you don't have good connection.
So,
You might just want to read it in the chat.
He put it in the chat. Okay.
Yeah, Martin, you're breaking up. You're in a bad zone.
sorry.
For nutrition yeah. Okay, Martin, I just read your question. We can't hear you very well, but I'll go ahead and answer that. So we have just begun looking at some of these nutrient biomarkers and how they relate to the CSF albumin index measure of blood-brain barrier disruption. So that's active work that's ongoing. What we have identified is that the ratio of plasma to CSF levels of nutrients is modified by blood-brain barrier dysfunction. Okay, so for example, vitamin C, which we are completely dietary dependent, has about a 4-to-1 ratio higher in the CSF than it is in the periphery. And as you have blood-brain barrier breakdown, you lose that concentration gradient. So the brain loses its ability to concentrate vitamin C in the setting of blood-brain barrier dysfunction. Now, what causes that? Whether it's the abluminal side of the blood-brain barrier is losing its integrity and the concentration gradient, and basically vitamin C is just flowing down its concentration gradient, or whether there is actually disturbances in the transport systems at the choroid plexus for vitamin C that becomes disrupted, and that's what's driving this effect is unclear. So I think it's important to look at potential nutritional modifiers of blood-brain barrier function, improving it, but also understanding that as the blood-brain barrier breaks down, the brain loses its capacity to concentrate nutrients that it has an innate ability to demand.
Any other last questions? Do you had something? Okay, we have a couple of more questions. So
Yeah, I think
do and
we
then
can pass to
Ariana.
Raja. Go ahead, Raja. Raja, go ahead. So
I think Ariana was first.
Okay. Uh, I was just going to follow up on, so you looked at like the brain barrier, but the gut barrier is also sort of a question here. And like there's been a lot of question about the interaction between diet causing gut barrier dysbiosis in people with AD, or maybe it's something about AD that's causing it. And so like potentially people, if they are remembering their diet correctly, they're still not getting these nutrients that would be coming from the diet due to the gut dysbiosis. So do you think there's something going on there on with that potentially in the nutrient profiles or?
Yeah, thank you for your question. This is also another really important area. So our approach is thinking a little bit differently. Instead of understanding what people are reporting, what they're eating, we're looking at profiles of nutrient concentrations and patterns of those that seem neuroprotective, and then coming up with a diet or regimen that would match that. Now, one of the potential challenges is the gut microbiome, leaky gut syndrome, these type of things. If you have that, if the setting has that, if you have that condition, whether or not changing somebody's diet is really going to get the profound effect is unclear. I mean, so this has had to be something that's addressed and identified before we would be successful in intervening and matching that plasma serum concentration. So what we're looking at here is kind of a few steps away. This is stuff that has survived and made it into the bloodstream. When you're looking at dietary intake, of course, one of the potential, as Stu says, the confounders of that reflection is the gut microbiome. Now, whether or not, those correlations that I showed between self-reported food frequency questionnaire and what's in the bloodstream, whether those correlation coefficients are modified by gut microbiome or leaky gut syndrome, is an important question to ask that we just haven't had. We only had 40 subjects in that study. But I think as we developed a brain nutrition lab at the McCance Center, it'd be great to have some interested scientists to pursue some of these important questions, because at the end of the day, that's going to be really key in terms of developing an intervention that's preventative in some form or fashion. So the relationship between the leaky gut and leaky gut syndrome and leaky blood-brain barrier, and this whole area and cascade related to nutrient delivery to the brain is right up our alley, but you can see how complicated it is. So I think I saw recently a wonderful paper on the reproducibility of gut microbiome measures that I think came out in Nature, but this is something we discussed in a 2013 Gordon Research Conference over a decade ago. So somebody has finally published the intra-individual reproducibility of microbiota species, which is really important, right? To extrapolate and develop. And I think that's been a real challenge with the reproducibility of that literature altogether. But I think now with that data, it can help you kind of hone in on what are the most reproducible measures and why, and start linking these different mechanisms together and extrapolate some next steps.
Gene, we're up at 12 o'clock here, but Roger, if you could ask a quick question, quick answer, so we don't pull people up.
Thanks for your question, Ariana.
Yeah, my question is very specific and very, very narrow, which is I saw a couple of your slides showing high correlation with fatty acid, particularly the palmitic acid, which I am very interested on. And that's not a trans fat, but a saturated fatty acid, and one of the major fatty acids found in the brain related to many neurodegenerative diseases. So is there any correlation that can be found to develop hypothesis based on fatty acid, saturated fatty acids?
Yeah, so we have not seen the saturated fats they've been neutral, Raja, in our studies, meaning the concentrations that we see. Now we're not doing feeding studies, right? I mean, a feeding study would be really the definitive study to determine their effects in living subjects. But in our observational work that has measured a lot of saturated fatty acid, including, palmitic acid, has been neutral. Some of the monounsaturated fatty acids that are very long chain fatty acids, like the 24-1, like nirvanic acid, that has popped up as something interesting. But I think a lot of the saturated fat work has been around medium chain triglycerides, using like C8 saturated fat or C10 saturated fat, and using that as a ketogenic agent that induces ketosis, and has seen effects over 6 months, meaning you can improve cognitive function over 6 months. Whether it works long term is unclear, and giving a saturated fat and the impact on biology from a vascular standpoint, whether that's safe as a long-term treatment, is also, there's a lot of debate about saturated fat in the diet. And of course, it depends on the type of saturated fat, but nevertheless, there are strong studies that suggest that higher saturated fat intake is associated with atherosclerosis and arteriosclerosis. So whether or not, giving a saturated fat, you have to get around that to develop your hypothesis, I think, in this space, given that vascular risk factors have been also predictive of Alzheimer's disease.
Great. Well, we're obviously a lot of interest in your talk, Gene. Thank you for an excellent talk, excellent Q&A, and thank you for all the questions for everybody. And I think we can end the seminar here. Any announcements about the next seminar yet from Gene? Or we got to be determined?
Yeah, we'll be in touch before the end of the year. But
Okay.
we have a strong Q1.
Okay. So this is, I guess, our last seminar before the new year, right?
Yeah, that's right.
Okay, and great. Well, happy holidays, everyone, and thanks again for joining us. Great job,
Thanks for
Gene.
attending.
Thank you very much.
Thanks a lot. Take care.